Have we have to fill some form?
Yeah, so there is this form that I'm going to send. Like I have already shared if people have not seen. So that form so you have learned what is a cell and you saw things under microscope, right?
Yeah.
In the previous session. So if you want to see anything around you or anything curious, how does it look under microscope? Just
Yeah.
fill it in this microscope, in this Google form. And depending on, if we can do it or not, we will uh, see and we'll show you in tomorrow's class.
Yeah. So have you posted in the chat?
Can you get the link?
Yeah.
So can you try opening it? Is it accessible now?
Yeah.
Yeah. Okay, anyone can't access it? Let me know.
Ma'am, it is still showing the same thing.
Yes, you need permission.
Why is it coming? Oh,
The form can only be by users in the owner's organization.
okay, or else just put it in the chat if you have anything to say. Or wait a second. What can be done? Question and answer. Can you see a question that I have posted? Can you see it? In the chat box?
We have to answer here.
Can you answer it?
No, ma'am, I don't see.
I can see it. What do you want to see under a microscope?
Bhatish, can you see it?
We have to speak here or we have to comment in the chat.
Okay,
But I could see it. Q&A.
Type anything in it, like as an answer to it.
Okay. An answer? How?
It's just a question. Sorry guys.
But where we have to type it? Where we have to type it?
For now, then just type it in the chat box.
Okay.
Just type if you want to see anything. Type it in the chat box. Is that fine? Now let's start with the session. Start sharing, start recording, filling. Can you see my screen? Can you see a phone camera?
Yes.
Yes. So do you guys have your phone with you now? Any?
Currently. Yeah.
You have?
Don't have a phone. My phone's already damaged.
Oh, okay. So there are few steps that you can do. So we are gonna do a called. Have you ever heard of the term augmented reality? What? Have you ever heard of the term augmented reality? Augmented reality.
Yes, ma'am.
Yes. So we are gonna see the cell with augmented reality.
Ma'am, you are not audible clearly.
Hello. Am I audible now?
Yes.
Yes, ma'am.
So go to your Google. So if you have a phone, can you see my screen?
Yes.
Yes. So type Google, type animal cell.
Animal.
So when you search, you would get this screen. Are all of you getting? This can be done only using phone. So those who don't have phone, I'll show you with my phone.
Oh.
So when you scroll down, can you see this something called view model?
Yeah.
Yes. So click and go to view in 3D.
I don't have 3D. Oh, this is so amazing.
So can you see the cell
3D.
rotating? You can move around the cell.
Yeah.
Those who can't, who don't have phone or don't have this option in their phone, see it through my phone. Is that fine? So
Yeah.
can you rotate it? Okay. So now there is this option called view in space, right? So click that view in space, and you are supposed to scan an area which is around you. And can you see the cell?
Yeah.
So this is exactly how the cell looks in real life. Can you see?
Yeah.
So these are all the different parts of cell that we learned today. So now tell me, what and all we learned? What is the purple thing in the center?
On the nucleus.
Nucleus.
Yes. What is blue color things that are surrounding the nucleus?
Lysosome? No.
The thing. We saw something is called highway of the cell. No, don't they look like roads?
Yes.
Ma'am, the
Actually, first I
endoplasmic
mean,
reticulum.
highways only came in my mind. But the
Endoplasmic
names are little
reticulum.
bit big, very hard to remember. Endoplasmic.
Reticulum.
Endoplasmic reticulum, smooth ear and rough ear. Actually,
Yeah,
rough
exactly. So can you see these red color dots? These are the ribosomes.
Yes.
Can you see?
Ribosome.
So yeah, some of them are attached to ER, some of them are not. Okay, and now you can see even go under and see. You know, these tube-like structures.
Yes.
Can you see?
Yeah.
You know, there are like these different tubes. Like it's a big structure. You know, these proteins can go inside this, and you know they travel to different places. And then we have here. What is this structure? Can you see? What is the structure?
Golgi apparatus.
Exactly, Golgi
Golgi
apparatus.
apparatus.
What are Golgi apparatus? They are post office of the cell, right? So whatever is coming from nucleus, they would go to ER, and then from ER they would go to Golgi apparatus. And in this Golgi apparatus, they are packed. Can you see the small balls? These balls. Can you see these balls? So these are like the package. No, those folds have certain structures which help them to produce these ATP. You remember we studied ATP? The electricity of the cell. The electricity that we want, the same thing the cells also want, and that is produced by this structure called mitochondria. Can you guys see? And then what else? What else did we see?
Vesicles.
Ma'am, the lysosomes.
Yeah. Someone asked something. Who asked?
Eric. It
Who
says
asked?
something. What is vesicles?
Okay, vesicles. So when you want to send
Vacuoles.
something, so if you want to send something to somewhere, what do? You pack it, right? You pack it into a package and then send it. So these vesicles are a package. So you can put whatever you want into this ball and send them wherever you want.
Okay.
Is it clear what are vesicles?
Okay.
And then you can see these ribosomes. Remember we studied ribosomes? These are the structures that help make all those parts. They are like factory machines. Can you see how many factory machines we have in our cell? And then what else? What else do we have? So these are something called centrioles. Can you see these things? These small things? Where is the cell? Can you see like the road kind of lines going all around the cell? Can you see the yellow color lines going around? Can you see the yellow lines, guys?
Yes, ma'am, we can see, but what is it?
So those are called microtubules. Microtubules. So these are like roads, basically. So if you want anything to be transported from one place to the other place, they would travel through these roads, and these roads are called microtubules. I'll show you probably in the afternoon session how molecules really walk on this road and go. They literally walk. I'll show you in a video. And can you see cytoplasm? Can you see the cytoplasm, the fluidy liquid that is surrounding everything? Can you see?
Yes.
Right. So that is the cytoplasm, and you can see the plasma membrane. Can you see the double layered plasma membrane?
Yes.
Oh, there is ER thing has some problem. So can you see now? This membrane which is going all around the cell, covering the cell. So this is the plasma membrane which will protect the cell from all the external things and will act as a gate. Interesting how cells look. Actually, in real life, complicated, right? Hello? Am I visible? Am I audible?
Yes.
Hello? Yeah. So what do you think now this is? Can you see these small X-shaped things in the center? Can you see these small X-shaped structure which is present inside nucleus? Do you know what they are? Anyone? If you don't know, just let me know. I'll tell you.
DNA.
Very good. So these are the DNAs which contain information. DNA. Very good. So is it clear? How many of you are able to open it in your phone?
Teacher, I could.
So it's cool, no? So all different cells have all these different kinds of complex structure inside them for their functioning. So it's similar to a city, right? A big city, with roads, with different places to go around and different roads. Interesting? You want to play some more? So there are, like, in this AR option, there are different kinds of related models. So if you just go down, you'll find. So this is a plant cell. Plant cell. Is it not coming? Whoever has phone can just go scroll down and there are a lot more options. Can you see? Like you can see mitochondria, you can see plant cell. I'll show you it. Is the screen visible, guys? Hello? Hello?
Yes.
I think you guys are playing with it.
Yeah, ma'am, it's a lot more enjoying.
Exactly. It's fun.
And the AR means how the AR is, no? They have made it. It's very like if you shake your phone, no, then also it is not disappear. It is very stable.
Exactly. It's a lot more fun to see things that you are studying actually in using such platforms.
And the thing is, I have touched here photosynthesis one, and they are all showing the life process that oxygen is releasing sunlight.
Oh, that's so cool. So can you see this plant cell here?
Yeah.
It's more complex than animal cell. And what is this big space that you can see in the center? These are vacuoles. So vacuoles store water in plant cells. So can you see the thick cell wall? Lot thicker than animal
Yes,
plasma
ma'am.
membrane, no? And see, almost all the structures are similar in plant and animal cells. So you have this
Ma'am, the one
ER.
thing which we can clearly see is not same is the vacuole. Like in plant cells, they are bigger vacuoles, and in animal cells, they are like smaller, and sometimes not even there are
Exactly.
vacuoles.
So you can see, in our animal cell, we didn't see any vacuoles. So you can see here chloroplast. Can you see chloroplast? So why do we have why do plant cells have
Ma'am,
chloroplast?
this is the pigment which adds the green color.
Exactly. So it has a pigment. This chloroplast is the organ that has the pigment chlorophyll. And this chlorophyll, that the pigment is called chlorophyll, and it helps in converting light energy to chemical energy. So plants need sunlight. No, what will happen if you keep plants in dark? If you don't give them sunlight, plants will die, right? So they need energy, and this energy is provided by these chloroplasts. So can you see? So almost all the structures are same in animal and plant cell, right? Except cell wall, chloroplast, and vacuole. Can you see the road kind of structure, the ER, the Golgi apparatus, the mitochondria? Can you see all the ribosomes? Everything is there here also. Can you see?
Yes.
Yes. Very good. Now, we have got, I'll close it. I'll close this session and we'll move on to the next set of experiments that we have. You guys want to play more with it? Or we'll move on. So you guys just roam around. So there are a lot more other things. So there is this bacteria, there is this mitochondria. Can you see? You have this mitochondria in close up. Can you see mitochondria?
Yes.
So this is the mitochondria. So you can see matrix, you can see ribosomes, you can see cristae. And this produces the energy currency called, what is the energy currency of the cell called?
ATP.
ATP, very good, adenosine triphosphate. Okay, and this is how the exact mitochondria looks like. And you have a lot of mitochondria in your cells, depending on the energy. So, our heart cells require more energy, right? Why? Why does our heart cells require more energy?
It
To
pumps
pump,
oxygen
to pump
to
because pump
all
regularly,
organs.
it stops.
Yeah, so it always beats very fast throughout our lifetime, right? So it needs more energy.
Yeah.
So I'll close it because my friends are waiting for the next session. They want to show you guys something. So I'll stop this. So is it interesting to see these things 3D?
Yes.
Yes, ma'am.
This means live
Have you guys
experiment.
ever seen it before?
No.
No.
No, give me. So you guys can kind of, if you have your phone, try exploring more other things that are there. There are bacteria, there are photosynthesis. It's very cool to see those things actually working in front of you.
Mitochondrion.
Give me a second.
Ma'am, have we have to left or have to join the next session?
So now two of my other friends would join you guys and take this session forward. So you guys stay. Like don't go anywhere. They both would join. So can you see Gopal? Kulkarni Gopal? He's one of my other friends. So they would be taking the next session. And that's a surprise. You guys would know when they
Yeah.
come.
Hi.
Good morning, sir.
Yes, sir.
You have applied.
It's blur. It's
Sir,
blur.
it's not my fault. Am I audible?
You can change it on the apply visual effects.
Yeah, now we can see.
Yeah.
Yeah.
Perfect. Continue.
Ah,
Yes.
so excited. Hi. Yeah, but you are a little bit of blur. Yeah.
Yeah.
Yes. Yeah. Slides, no? Oh. No.
Little glasses. Glass like rectangle. Slide.
Oh,
Even blood cell, leaf, everything.
Yeah, I have seen
Yes,
it
sir.
before in videos, but I have not been that heard that name. Oh, yeah. Yeah.
After putting the sample blood, then we put another slide on the top. Sample. Yeah.
Yeah. Yeah.
Yes, Didi, in our school lab, we had also seen in the microscope, and we had actually like first examined it from the outside, and I had seen these the rectangle glass side and the square glass side.
Yeah.
It's from a colored
Yeah, yeah.
type of.
Because they are sometimes transparent. Because it highlights the cells. Only cells it highlights.
Cover slide.
Cover slip. Cover slip. Yeah. Yeah.
It's just water vapor.
Yeah.
They are just air bubbles.
Guys, can you pin your screen? Gopal, Devshish, just pin your screen.
Hello. Hi. Hi.
Hello.
Hi. Yes.
So these are at the different chemicals that we use. Can you see this big bottles?
नहीं नहीं वो उसका
Oh, yes.
म्यूट कर दो उसको म्यूट करके
Yeah. So this is like our lab space. This is where we work in our college. There are like different machines. So we have fridge. We also have fridge. You need to store a lot of microorganisms under 4 degrees Celsius. So we store all the different chemicals. really came up with the idea of these cells and all, right? Do you know who was the first person to see the cells?
No.
Yes. So let's look into the history of cells.
Actually, ma'am, history is interesting, but the worst part is its names.
You don't really need to know all the names, like once um, you just need to know that these people have spent their entire life searching for it, and how awesome it is. They want to search something, they found something,
Yeah.
and that's why we are here doing so many research and fighting COVID and all. It's all because of these small historical people's wonders that they have done. So let's move. So we have our first person in the history book of cells. We see Robert Hooke. So almost in every textbook you would find his name, Robert Hooke. So he was the one who preliminarily designed the microscope that you saw today. You saw, right? There were two lenses. You will
Yes.
see from the top, and
Yes.
from the bottom the cells would be zoomed in and you would see it. So this thing is called compound microscope, and he was the first person to design this. And he actually observed these cork cells. You know what is cork, right? So there is this bottle. So when you see movies and all, there'll be this message bottle, and they'll close it with a cork. Have you guys seen what cork is? Hello, am I
No.
on? You guys don't know what a cork is? So cork cells are like, it would be very similar to plant cells, but they're dead. They're not living cells. So they're like this outer bar kind of. Can you see? Can you see these cells?
Yeah.
So this is something what he observed. So this is what he drew. He drew this in his paper. So that those days we didn't have a photo camera and all, right?
Yeah.
So this was drawn by him, what he observed. So it's very important to, record things, whatever you do. So do you guys have the habit of keeping diary writing?
Honestly, no.
Even I don't, but it's good to keep the habit of writing a diary.
Actually, I used to write a diary. I finished it.
Uh-huh. You're not starting for this year?
I used to write the story. I now not.
So all these famous history people, we know their work, what they did, only because they had good record keeping. So whatever they used to see under microscope, whatever they used to observe, they used to write it down. Okay, this date, this is what I saw. This date, this is the structure that I saw. So everything would be kept in a book, and that book we can see now. So that's how it's important, record what we see in like at least lab work. So he was the first person to call it cells. So they look like these small box kind of cells, the prison cells and all are there, right? So it was looking like this blocks. So he called it cells. So this was the first guy who found a microscope and saw under the cell these small cell, and he was like, okay, these so many living organisms around us are made up of these small things called cells. And this is the microscope that he designed. Can you see?
Does that microscope today also works?
This microscope?
Today also works?
Yes, it works. It has a very basic, and a strong fundamental principle. So it works today also. And do you know these glass-like things? Why are they here?
No.
Can you see a flame here?
Yes.
Yes. So what did I tell you when we were learning? Microscope needs light, right? You
Yeah.
need light to see things. And
Oh, I can see a lens which points at that.
So those days for focusing light, they didn't have anything. So, when the flame is burning here, you'll get light, right? So this glass ball, what does it do? It will point that flame light towards this circular thing. Can you see this circular thing? So
Yes.
whatever flame is there, this glass ball would kind of focus it towards this place. So what happens? This is where the sample is, right? So the sample would be brightened up. Now we have torch, right? When you have torch, so what happens? The light comes only from that hole. But those days they didn't have torch, so they used this technique. So this technique, this ball tries to make a torch of this flame light, the light of these flames. And through this hole, you can actually see. So these holes have these lenses. Awesome, right? Even at those age, someone made this much sophisticated microscope, and for the first time, he was able to see this.
At what time he had designed or built it?
1600.
1665?
1664. So that is how many years back? How many years is back?
More than 650 years.
Exactly. Not 600. I think, yeah, 600. Yes. I'm so sorry. I'm still living in 2020 when the corona struck. Yes. So it's, I don't think it's more than 600. Right? It's 2022, right? 400.
Yes.
Around 400 years ago. So there was
yeah.
a man who was exceptionally brilliant, and he found this. I don't think like the historians, they really had this eagerness to see things. They had so many creative ideas, and they created so many machines so that they can explore things out. And that's the same thing that we do in our college also. We'll try to explore things, how this works, how that works, what is causing it. That curiosity should always be there, in all the kids. So next we have our another person, Anthony van Leeuwenhoek. It gives me a very Harry Potter name feel. Anthony van Leeuwenhoek. So he was basically a merchant. He was not even a scientist, or he was not even related to science, So he was simply a merchant who used to sell clothes. So to know how much quality his threads have or whatever cloth he's making, he used lenses. So most of these people who discover these cells, microbes, they used to love playing with lenses. They used to get this glass piece. They used to see different objects. We also tried doing that, no? Like when you get a magnifying glass, you go see different things. How this would look in a zoomed in version. We all have that, curiosity to see things that we can't see. The same curiosity these people also had. So through his lens, you could see he observed these threads and these components very big. And this person, Anthony van Leeuwenhoek, was the first person to observe live cells. So the Robert Hooke, he observed dead cells. So Koch cells are actually dead. But this person, he was the first one to observe live cells. And he was the first person to actually observe bacteria. Bacteria, right? Microbes. So this discovery or his discovery of this bacteria opened up a whole new world of microorganisms to the people. So they were like, oh wow, there are like these small, minute things roaming around all around us. And all of them got so excited and curious to know more things about these small, little, microorganisms. Because before this, they don't know. They were like, they didn't know that such things even exist. And after him observing this bacteria and live cells moving, he was more interested. Okay, what are these cells? How does it look like? How many different types can we find? And this is the Leeuwenhoek's microscope. So this is the microscope that he made. And it is not that sophisticated as Robert Hooke's, but he made it a single lens. And he used to see like this. He used to hold and see it through that small pinhole. Can you see here? Hello, am I audible? Hello.
Yes.
So he used to see through this, and he observed a moving live cell. then you have different people, all the different people started, you know, observing more cells. Like they used to take water sample, they used to take different plant cell, animal cell, and they started seeing all the cells under microscope. And this is when Robert Brown, he discovered or he observed opaque areas in cells. So when you saw in microscope, you saw a very dark stained region, right? The center part of the cell alone was stained very dark.
Yeah.
Yes. So those places were called nucleus. And this person also observed the same thing. So he was like, oh wow, like all the cells, somewhere there is this dense part which is staining a lot. And if you see under microscope, it is not very transparent. It has something in it. You know, when you have transparent things and when you have opaque things. Opaque means it's not transparent. You have some material, lot of material in it. So he was like, okay, there is something, some non-transparent thing in one particular area of a cell, and he called those areas nucleus. So he was the first person to name that condensed area nucleus. And then let's move on. So these are like different people who came up with different theories. So once people started, you know, seeing different cells, you know what is a theory?
Yes, I know.
What do you call a theory? What is it? So theories are like these set of behavior or function that all the set will follow it. So if we say gravitational theory, that means if you drop an object, it will fall. So it is accountable for all the things in Earth, right? If you drop a ball, if you drop a elephant, if you drop a apple, everything falls down. So it's a theory. So these people also came up with few theories after seeing some numerous cells. So they were like, oh, all these cells have the same behavior. All these cells not look like they show same function, or they have certain things that all of them have in similar. So those are something called cell theory. And it states all living things are composed of cells. Is that true? You guys have seen so many photos and videos and microscope today, right?
Yes.
So is there any non-living thing that is made up of cell? We don't call it living.
No.
We don't call it living. If something is not made up of cell, we don't call it living. But there is a very good controversy of virus being living or non-living. That would be told by you today in the afternoon session. So when we go through to microbes. So in general, all living things are composed of cells. Now, what is the next thing? Cell is the basic unit of life. So every life, it starts from one cell. So one cell multiplies million, trillion, zillion, and that's what builds a human body or an animal or a plant. That cell is the basic unit of life. It's a living cell, no? We saw the components of cell and what does it do, right? So it has nucleus, it has energy, it has energy making mitochondria, it has highway, ER. So it is like the basic unit of life. And the third point is that new cells arise from existing cells. So what does this say? Can anyone guess? What does this statement say? What is your opinion? Comment on this sentence. New cells arise from existing cells. Anything, guys? It's totally fine. What do you understand by this sentence?
The about new cells, the inventor of the cells, discovery of cells. that who discovered this, who made microscope, um, the basic unit of life, that about new cells arise from existing cells. Sorry.
Yeah. So new cells arise from existing cells means that, when we are born, our mother gives birth to us, right? Like we are born due to, like, our mom has given birth to you, me, everyone. Similarly, only if there is a cell which is already present, it would give birth to the next cell. It can't appear out of air. You can't say that God created cells. It can't appear out of nowhere. So that is
This is that okay, that one cell gives birth to the next cell, but what about the first cell on the earth?
Yes, good question. Very good question. And this comes under a topic called evolution. So the first cell that came, okay, it didn't have a previous cell, but it had something similar to a cell. So how a cell was first made, there were few components. So there was this soupy thing that you call, like when you learn evolution, the earth was, very hot ball of some planet. And then it rained for some 1,000 years. Have you guys seen this evolution story? Like, have you guys seen any video
Yes,
or any?
little bit.
Yeah, so around for some 1,000 years, maybe more than that, it was raining heavily and continuously. Our earth was like this soup of, it was hot, and then it was falling, like rain was falling. And in this water, what happened? Like there were few chemicals, some molecules that were there. They started forming something called a boundary, and they started remaining in groups. Like they formed like a ball kind of structure, and they were like present all over the place. And then they adapted, and then they got some more proteins into it, and they kind of started functioning like a cell, but it's not still a cell, like a cell. And then they gave rise, they again adapted, and then they had some more components. So it's not like, it came out of nowhere. It is like a work of some 100 different processes. So they evolved, they changed themselves according to their environment. So if they needed, so that time it was carbon dioxide. So the entire atmosphere was filled with carbon dioxide. So can we survive in an atmosphere full of carbon dioxide?
No.
No. Yes. But in those times, the condition was like that. The entire atmosphere was full of carbon dioxide. So the cell or the cell-like things that time were totally different. Then when oxygen started to be produced, the cells changed. They got new things into them, and then they got chloroplasts, they got mitochondria, so that they can make use of this carbon dioxide and give off oxygen, or take oxygen and start making energy molecules. So it's like a very, lengthy process of, a cell being made. So once, so it's not exactly, we don't have anything and suddenly a cell came. So there was some cell-like ancestor from which one component was added and it became a proper cell.
So if we study the origin of cell, then probably can we make a cell?
The thing is that we don't have enough knowledge about cell itself.
No, that is okay. But if we somehow in future, if we get to know the origin of cell, then can we able to make?
Yes, of course, you can make. But I feel you still need a pre-existing. So for now, people make cells from pre-existing cells. But so now they make cells from pre-existing cells. So we have cell and they make a cell out of it. But completely a new cell from scratch, we can possibly in the future. because you still need to know what you define as a cell, because cell is very complex. Like what you studied today is like very basic things. It has lot, like, so one component will affect another component, and when you talk about this DNA thing itself is like some ocean level information that they have. So creating another new cell means you're giving that information booklet also. So what information booklet you'll give to them? Will this cell be able to survive that information book? So that information booklet is basically DNA. So what DNA you're going to give them? So it all comes under evolution. But yes, it's very interesting, if you can make a cell. But it's also very dangerous. You can make any type of cells, no? Then it has both its ethical issues, and also when you start any experiment or any project, there are a lot of organizations which check whether your experiment or your thoughts is it going to harm anyone any time or like that.
Yeah.
Really cool, science, seeing things, learning things, how things work.
Yeah. Except the name part, I love biology.
You would even enjoy names if you learn it in the proper way. Just ratamana, like just mugging up things, it's boring. Mugging up things is boring. You need to, enjoy when people came up with that name. So a lot of names have this, meaning behind it. Why did they name it? So you can correlate that. You know, why is it named like this and all. So it's fun to learn that way. Okay, guys. So can we join then at 2:30 then?
The same link to at
Yes,
2:30.
the same link. So you guys can
Yeah.
have your lunch, take a break.
Yeah.
Bye.
Bye.
So we should join at 2:30 or 2 o'clock?
2:30.
Okay.
Goodbye, guys.
Bye. 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 Bye.
Bye.